Cell, the unit of life. Introduction. What makes an organism living? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. What is a cell? Cell is the fundamental structural and functional unit of living organisms. Anything less than a complete structure of a cell does not ensure independent living. Anton von Leeuwenhoek first saw and described a live cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which forms the tissues of the plant. At the same time, Schwann, in 1839, a British zoologist, reported that animal cells had a thin layer called plasma membrane. He concluded that plant cells have cell walls. Schleiden and Schwann together formulated the cell theory. But this theory did not explain as how new cells are formed. Rudolf Virchow explained that new cells arise from pre-existing cells. Omnicellula is cellular. And finally modified the cell theory as all living organisms are composed of cells and products of cells. All cells arises from pre-existing cells. An overview of cell. Cells differ greatly in size, shape and activities. For example, mycoplasma is smallest cell, while egg of an ostrich is the largest isolated single cell. Nerve cells are some of the longest cells. The cytoplasm is main arena of cellular activities in both plant and animal cells. Ribosomes are non-membrane bound organelles found in both eukaryotic and prokaryotic cells. Apart from cytoplasm, they are also found in mitochondria, chloroplast, and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. Cells that have membrane-bound nuclei are called eukaryotic cells, that lack a membrane-bound nucleus called prokaryotic cells. Prokaryotic cells lack membrane-bound cell organelles, are represented by bacteria, blue-green algae, mycoplasma or PPLO. In addition to genomic DNA, many bacteria have small circular DNA outside the genomic DNA called plasmids. Plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. Plasmid DNA is used to monitor bacterial transformation with foreign DNA. All prokaryotes have a cell wall surrounding the cell membrane, except mycoplasma. Cell envelope and modifications. Most prokaryotic cells have cell envelope, which is tightly bound three-layered structure. The outermost glycocalyx followed by cell wall and then the plasma membrane. Glycocalyx may be a loose sheath called slime layer or thick and tough called capsule. The cell wall prevents bacteria from bursting or collapsing. Extension of plasma membrane into the cell in the form of vesicles, tubules and lamellae are mesosomes. It helps in cell wall formation, DNA replication, distribution of daughter cells, respiration, secretion process and increase the surface area of plasma membrane. In cyanobacteria, chromatophores contain pigments. Each layer of the cell envelope performs distinct functions. They act together as a single protective unit. The plasma membrane is selectively permeable in nature and interacts with the outside world. It is structurally similar to that of eukaryotes. Bacteria may be motile or non-motile. If motile, they have flagella composed of three parts: filament, hook and basal body. Pili and fimbriae do not play a role in motility. Bacteria, on the basis of the difference in the cell envelope, can be gram-positive or gram-negative. Ribosomes and inclusion bodies. Ribosomes are 70S, has subunits 50S and 30S. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. Ribosomes are associated with the plasma membrane. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material is stored in the form of inclusion bodies in prokaryotic cytoplasm. Example: phosphate granules, cyanophysin granules and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Eukaryotic cells. Besides the nucleus, eukaryotic cells have other membrane-bound structures called organelles, like endoplasmic reticulum, Golgi complex, etc. The eukaryotes include all the protists, plants, animals and fungi. Plant cells have large vacuole. Animal cells have centriole, which are almost absent in plant cells. Ribosomes are of 80S in cytoplasm. Small subunit is 40S and large 80S. Cell membrane. Chemical studies on the cell membrane, especially in human RBC, enabled scientists to deduce the possible structure of plasma membrane. Cell membrane is mainly composed of proteins and lipids, mainly phospholipids. Phospholipids consist of polar head, outward, and non-polar tail, hydrophobic, inner side. In human RBC, 52% is proteins and 40% is lipids. Membrane proteins can be integral or peripheral. Most accepted model for structure of cell membrane is fluid mosaic model given by Singer and Nicholson in 1972. Membrane is selectively permeable. Many molecules can move across the membrane without any requirements of energy is called passive transport. Movement of water by diffusion is called osmosis. Many molecules require energy or ATP for their transport called active transport. Example, sodium potassium pump. The quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. Polar molecules cannot move through the non-polar lipid bilayer. The fluid nature of membrane is important for functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. Cell wall. Non-living rigid structure called the cell wall forms an outer covering of the plasma membrane in fungi and plants. The cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on inner side towards membrane of the cell. The middle lamella is a layer mainly of calcium pectate. Algae have cell wall made of cellulose, galactans, mannans and calcium carbonate. In plants it consists of cellulose, hemicellulose, pectin and proteins. Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as a membranous system because of their functions are coordinated. Endoplasmic reticulum. Reticulum of tiny tubular structures scattered in the cytoplasm is called endoplasmic reticulum. The endoplasmic reticulum has ribosomes on surface are called rough endoplasmic reticulum. In absence of ribosome, they appear smooth called smooth endoplasmic reticulum. A rough endoplasmic reticulum is involved in protein synthesis, whereas smooth endoplasmic reticulum is involved in lipid synthesis. Golgi apparatus. These were named Golgi bodies after the discoverer named Camillo Golgi. They consist of cisternae which are concentrically arranged near the nucleus with distinct convexes or the forming phase and concave trans or the maturing phase. Principally performs the function of packaging of materials. It is the important site of formation of glycoproteins and glycolipids. Lysosome. These are membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. They are rich in hydrolytic enzymes, lipases, proteases and carbohydrates, optically active at acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids. Vacuole. The vacuole is the membrane-bound space found in the cytoplasm. Membrane is called tonoplast. Contain water, sap, excretory product and other material not useful for the cell. In amoeba, the contractile vacuole is important for excretion. In many cells, as in protists, food vacuole are formed by engulfing the food particles. Mitochondria. Mitochondria, unless specifically stained, are not easily visible under microscope. Each mitochondria is a double membrane bone structure with inner compartment called matrix. The two membranes have their own specific enzymes. The outer membrane forms the continuous limiting boundary of the organelle and inner membrane forms cristae. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence called the powerhouse of the cell. The matrix has a single circular DNA molecule, a few RNA molecules, ribosomes 70S and the components required for synthesis of proteins. Here S Sudberg units stands for sedimentation coefficient. It is an indirect measure of density and size. Mitochondria divide by fission. Plastids. Plastids are found in all plant cells and in euglenoids. Based on the pigments, plastids can be classified into chloroplast, chromoplast and leucoplast. The chloroplasts contain chlorophyll and carotenoid pigments. The leucoplasts are colorless plastids. Amyloplast stores carbohydrates, example potato. Elaioplast stores oil and fats, whereas the aleuroplast stores protein. Chloroplasts are double membrane bone structure which has membranous sac-like structures called thylakoids, and the matrix is called stroma. It also contains small double-stranded circular DNA and ribosomes. Carotenoids is fat-soluble pigments, example keratin, xanthophyll, etc. The ribosomes of the chloroplast 70S are smaller than cytoplasmic ribosomes. Thylakoids are arranged in stacks called grana, singular granum, flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. Stroma contain required enzymes for carbohydrate and protein synthesis. Chlorophyll pigments are present in the thylakoids. Cytoskeleton, an elaborate network of filamentous proteinaceous structures present in the cytoplasm, is collectively referred to as the cytoskeleton. It is involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from eukaryotic flagella. The central core is called axoneme, and the arrangement of axonemal microtubules is referred to as the 9+2 array. Both cilium and flagellum arise from centriole-like structure called basal bodies. They are covered with plasma membrane. Plasma membrane, peripheral microtubules, doublets, interdoublet bridge, central microtubule, radial spoke, central sheath. Section of cilia and flagella showing different parts. A is of electron microscope. The next one is diagrammatic representation of internal structure. Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials and lies perpendicular to each other. The central part of the proximal region of the centriole is proteinaceous called the hub, which is connected with the tubules of the peripheral triplets 9 by radial spokes made of protein. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of nucleus was given the name chromatin by Fleming. Interface nucleus has chromatin, nuclear matrix and nucleolus. Nucleus has the membrane and the space between two membrane in perinuclear space. Outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. During different stages of cell division, cells show structured chromosomes. Chromatin contains DNA, some basic histones, some non-histones and some RNA. Every chromosome has primary construction called centromere, on the sides of which disc-shaped structures called kinetochores are present. Based on the position of centromere, the chromosome can be classified into four types. Metacentric, centromere is in the middle. Submetacentric, centromere slightly away from the middle. Acrocentric, centromere situated close to one end. Telocentric, centromere in terminal position. Sometimes a few chromosomes have non-staining secondary constructions at a constant location. This gives the appearance of a small fragment called a satellite. Nucleolus is not a membrane-bound structure and it is a site for active ribosomal RNA synthesis. Types of chromosomes based on the position of centromere. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells.